Using the Spotiton robotic system, one can mix and vitrify a protein of interest with an interacting partner on an electron microscope grid in as fast as 90 milliseconds. This protocol allows the capture of intermediate protein conformations that are too transient to be captured by standard grid preparation techniques. Time-resolved Spotiton can inform sub-second biological or biochemical systems, such as membrane channel activation, DNA or RNA synthesis, or early interaction of a drug or antibody with its target protein. The user must concurrently manage several components that directly impact the quality of a grid. It is important to understand the system before using and have patience when preparing grids.
To begin, open the main valve on the nitrogen supply tank and ensure the system reservoir is filled with degassed, ultra-pure water. Turn on the computer and the Spotiton system at the multi-outlet power strip. Click on the desktop icon to open the user interface of the Spotiton software. In the Tools menu, select Initialize Stages to initialize and home the 3-axis robots and rotating dispenser head assembly, ensuring the dispenser tips are pointing down prior to initialization and homing. On the main menu, click on Go to Safe Position to send the robots to the safe position. On the Aspirate tab, select Prime to flush the dispenser heads multiple times with water from the reservoir. Continue until 2 uninterrupted streams of water can be seen emerging from the tips. On the Inspect tab, send tip 1 to the inspection camera and test fire water, adjusting amplitude until discrete droplets are produced.